you may remember, well, you don't remember because I haven't told you yet, but the assignment, first assignment we're going to have for biobanking is going to be on, surprise, surprise, design your own biobank, right? So I'm going to present to you today a real biobank from my own research that I've been working on. And basically, we're going to go into the different components. So the first component when you want to think about a biobank is obviously the experimental design, right? You want to understand, the science and the purposes for which that specific resource was developed in the first place, right? So you're not going to spend millions of dollars developing a resource unless there's some specific clear application behind it. And so I'm going to give you my own experiences in developing one of the first Latin American biobanks, which is called the Peruvian Genome Biobank. And then I'm going to give you some details of a proposal that I submitted that hasn't been successful yet, but it gives you an idea of the stuff that you need to think about and how much it costs. Okay. So also very much connected to one of the global health priorities that we have today, which is, the fact that non-European populations are severely underrepresented in the sort of global landscape, global data landscape. And that has tremendous implications in our understanding of the specific manifestations of diseases in those regions. So as usual, like you see that with large language models such as ChatGPT and so on, they're incredibly biased towards the North American, US-centric worldview. And the farther you are from that sort of framework, the less relevant or the more hallucinations they have. So something like that happens in biomedical research, because most of the data is based on Northern Europeans. Then the further you go from that sort of framework, the less accurate the models are going to be that represent our understanding of health. So I'm going to focus today on, for me, the Latin American continent, which is something I've been working now for about a decade. The fact that I'm a native Spanish speaker helps, I can tell you that. And that's precisely why I think that has given me a strong advantage, because, I can serve as a sort of bridge between the amazing things happening at biobanking level here in the UK and the Spanish-speaking world in Latin America. And so that has sort of given me a strong advantage, because to my surprise, in Latin America, unless you speak either Spanish or Portuguese, it's very difficult actually to really interact. I'm not sure how that happens. What's the case in Africa? I mean, Africa is mostly English and French, right? And people, if you speak English, you should be all right in Nigeria, right? That's not the case in Latin America. Unless you speak Spanish, it's very difficult, I found. And people tend to be very sort of close. And I've always, even though I'm myself Spanish, and I've tried to sort of, telling them, guys, you need to move on to Spanish. The scientists, the doctors, there's a reluctance somehow to get into the English speaking world. So this community actually is no longer invisible in London. In fact, the Latin American diaspora in London, bigger. So the Latin American diaspora in London, is that good? Has now is grown by 400% in the last 2 decades. So unfortunately, though, it's been reported that around 40% experience workplace abuse and exploitation, and around 10 times average are illegally paid below the minimum wage, right? So that's a clear sign of inequity. Whereas some big universities, such as Queen Mary University of London, have devoted strong efforts on characterizing health outcomes for the British, Bangladeshi, and Pakistani people, with a very strong investment on these communities here in the UK. That hasn't happened for Latin American communities. And I, as a consequence, would like to frame the remaining of this presentation around research in Latin America with these three following statements. And every time I talk about indigenous genomics involving this research, I always bring up these three statements on how I see ethically how my research should be conducted. So first of all, I would like to express my respect for peoples and traditions. That means we follow care principles, which stand for collective benefit, authority to control, responsibility, and ethics. So I fully respect indigenous peoples and their rich traditions. The work that you're gonna see here with indigenous data has been conducted with the utmost sensitivity and acknowledgement of their cultural significance and heritage of their civilization. And I strive to honor their legacy and ensure that my research upholds the dignity and respect owed to their descendants. I condemn genetic colonialism. By genetic colonialism is basically many practices in the past where scientists from the West will go collect the samples and then forget about the local peoples and not basically involving them in the process of serving their needs. So I unequivocally condemn genetic colonialism in all its forms. Have you ever heard that expression, genetic colonialism? Well, that exists, okay? You can imagine what that is, right? It's a sort of 21st century colonialism, slavery, whatever you want to call it. So my research is guided by a commitment to ethical standards and practices that respect the autonomy and rights of indigenous populations. And when I say indigenous population, I can say people from Kazakhstan, people from Pakistan, people from Nigeria, Yoruba, whatever you want to call them. And I'm dedicated to ensuring that the benefits of research are shared equitably with the communities involved. I also recognize that the historical and ongoing distrust in science among indigenous populations continue. And I cannot take for granted that me being a scientist or us being a scientist are going to necessarily going to be trusted. So we need to earn the trust of peoples. Therefore, the way to do that is by building bridges through transparency, collaboration, and dialogue. And so I aim to contribute positively to the relationship between science, indigenous peoples, and promoting trust and cooperation. Right, what do you think of that introduction? Any comments so far? You didn't expect that, did you? No? It's interesting, right? Yeah. That's a sort of, hopefully, a way in which we should frame our research for this kind of work, right? And this is can be extrapolated to any biobanking, any data that comes from research participants. What do you think? You agree?
Yes.
Do you like it?
Yes.
Yeah?
We have indigenous I'm from Malaysia, so we have a lot of indigenous populations as well. Who have gone through a lot of issues as well, not only from the time when the whole country was colonized, well, a massive part of the country was colonized, and now with the current government, there is a form of colonization over the indigenous people as well. So even though we don't have our own biobanking efforts yet, but I certainly connect with
You will.
with, yeah.
You know what? Because biobanks are one of the greatest resources for the country. And I'll show you, hopefully at the end of this lecture, you'll know why. You'll be able to see why, okay? Now, let's talk a little bit about historical context in Latin America, right? And that brings me to the years around 1,400 of our current era, okay? So you can see there the Inca Empire, also known as Tahuantinsuyo. It was the largest empire in pre-Columbian America, and its administrative center was located in Cusco, present-day Peru. So at the height of the empire, you see there, it extended across the western part of South America, encompassing various environments from the Pacific coast to the Andes and to the Amazonian rainforests. So the peopling of South America happened no later than 23 years ago, when the ancestral peoples from East Asia crossed the Bering Strait and then came down the coast through South America, and then through the Panamites moves into South America. And because of the geography, you have like three main sort of geographical regions there. One is the coast, one is the Andes, another is the Amazonian rainforest. And you can see how human evolution mimics those geographical patterns. So the first human colonizers, Homo sapiens colonizers, they are believed, we can estimate that they reached South America around 12,000 years ago. And although there was these three branches and some, we call it admixture between the three, I also want to bring to your attention the fact that you can actually reconstruct human migration patterns just by looking at their genomes, at the DNA. So, which is, I think, incredible that we can do that. So the Inca came along around the 1,300, then they grew, and then on 1532, my ancestors touched land for the first time in 1532, okay? So there you have my lovely ancestors. Some of them, some people say they don't like them. So I'll try to be as transparent and as faithful to the truth as I know it. So the Spanish conquest of the Inca Empire started in 1532. So you can see here Francisco Pizarro, who was born not far from where I'm from, in southern Spain. And their arrival had devastating effects on the indigenous populations, because they inadvertently introduced a number of diseases that the populations had never been exposed to. So while influenza was not as deadly as smallpox, it did also cause significant deaths among native populations. So there were a number of catastrophic epidemics that swept the region, even killing the Inca leader Huayna Capac. You can see him there, and his son. And so these waves of epidemic disease included the smallpox, influenza, measles, mumps, dysentery, typhus, and pneumonia. So you can imagine. And this is what happened to the Inca population. So in the span of 40 years, from a peak of around 16 million, they were reduced to less than 2 million. Okay. So the pressures that this made into the population, this was basically a huge population bottleneck, as you can see, right? And the effects are seen today. As even today, we still see the effects of that. So if we look at the Native American ancestry proportion of the population, we see that Guatemala is quite strong on that, but so is Peru and Bolivia and Greenland. So, but the majority of people I would say, correct me if I'm wrong, in Latin America, they're admixed. So they have a little bit of indigenous, they have a little bit of European, a little bit of African, a little bit mixed. It would be interesting to see your genome. Probably 60%, I'm guessing 60% indigenous, 40% Spanish. Yeah. Something like that. Yes. Right. So now what this means is that the things that have been developed for Europeans don't necessarily work for these populations. And in fact, if we look at the indigenous Americans, what the representation is across the world, they're among the least well represented in any kind of biobank that you can think of around. So you can see these are we call it the GWAS diversity monitor. You may have heard of GWAS, Genome-Wide Association Studies, which are the workhorse model for genetic modeling of complex diseases. So the diseases that people die of, that is, diabetes, cardiovascular disease, neurological disorders, they're all complex diseases. By that, I mean many genes contribute to the susceptibility to that risk. Unfortunately, Kazakhstan also looks quite empty. And since it's a big country, then you have a big blob there missing. Interestingly, so I've talked to some of you in our one-to-one conversations about something called pharmacogenomics. I'm going to remind you what pharmacogenomics is. So pharmacogenomics is the study of how genetics influences your optimal dosage for any drugs. So all of the drugs, the 400 odd drugs that exist approved by the FDA, which is the main Food Drug Administration, the main sort of one of the leading legal entities that approves and regulates the sale of medicines. They're mostly being developed for Europeans, okay? And so that's an issue. So now, Alina, I have a question for you. Would you consider Kazakhstan is South Asian or Central Asian? Right. So according to PharmGKB, now it's been called Clean PGX. Some of them, we had a look look at it. So this database that collects all of the pharmacogenomic data sets into one single location called Clean PGX classifies Kazakhstan as South Asian. And apparently, African is now divided into Near Eastern, including Morocco, down to Iran, and then Sub-Saharan African. So in other words, you can see that the boundaries which are used to classify global populations in pharmacogenomics are completely arbitrary, i.e., rather than based on biology, they are based on politics. And that has tremendous implications because, the differences, even in the US or in the UK, there are people from African ancestry, there are people from European ancestry. You can't, even in Africa, the genetic distance that exists between neighboring tribes can be as big as the differences that exist between Europeans and Chinese. There's more genetic variation within Africa than the rest of the world combined. Can someone tell me why that is? No, but thank you. Because Homo sapiens originated in Africa around 150,000 years ago. And it only came out of Africa around 80,000 years ago. So there has been a lot more time for Homo sapiens to evolve within Africa, and hence more time for accumulating differences in the genetics. And only a very small subset of Africans migrated outside of Africa and then started colonizing little by little the rest of the world. So that's why there's so much more diversity in Africa than outside of Africa, because there's been a lot more time of evolution of Homo sapiens. That, as I said, and then, so it came out around 80,000 years ago outside of Africa, and as I said earlier, they crossed the Bering Strait 23,000 years ago. So the indigenous populations in the Americas, the oldest will have been the 23,000 years, right? But of course, we're talking at that time, 23,000 years ago, we had an ice age. So obviously the sea would have been frozen. They would have been able to cross. The UK, for instance, at that time was connected to Europe. So I mean, as you can see, geography has a very strong impact in how these migrations occur. And as a consequence, the adaptations to the environment that have had as a consequence the inheritance of specific traits that some other populations might not have had, right? So in here, on this side, on this bottom half of the presentation, I'm showing you the proportion of data for which we have gene-drug reactions understood or studied from the total. And as you can see, around 63% are European origin, around 26% East Asian, which is mainly Chinese and Japanese. And then the rest of the world are virtually underrepresented. Any questions? Any comments? What do you think? Have you learned anything new that is useful? Yeah? Not so much? Okay. So the Peruvian Genome Project started first with the collection of 100 participants that were distributed across 7 different locations in Peru. Although Peru is just one country, it's one of the most diverse outside of Africa, is one of the most diverse countries in terms of different populations in the world. And it's because it has, on the one hand, you have the coast, which is one of the most arid regions in the world. So it's completely desert. Then you have in the middle the chain of mountains. Around 30% of the population in Peru live above 3,000 meters high. And living at 3,000 meters high means that you have the equivalent of 60% the same amount of oxygen as you would have at sea level. So in other words, there's the equivalent of less than 40% oxygen. And the ultraviolet radiation is, I don't know how many times, but I can tell you, I was there for a couple of days, and I was absolutely, with 50 factor, I was burnt. Whereas the people there, this is the Uros who live on Titicaca Lake, which is 4,000 meters high in here. It's not just that they don't have sun cream, but and the high altitude of ultraviolet rays, but it's also the reflection on the water as well. It's like when you go skiing, that you have that extra sort of exposure to ultraviolet. And I was wondering, why do they not get melanoma? Why do they not get skin cancer? And, an European would be there, half a year, and they get cancer straight away. So obviously, there's a lot of biology there that we still don't understand that could help us really beating diseases, simply because we have this natural experiment that has occurred throughout millennia, where through adaptation, through evolution, you have these populations which are now adapted to that circumstances. And as a consequence, they must have biochemistry that allows them to thrive at hypoxic level. If you are at hypoxic level, usually your immune system, correct me if I'm wrong, your immune system would trigger an inflammatory response. They don't. But then the other side of the coin is that they have a high susceptibility to diseases such as tuberculosis. So we know that some of these populations, you take them outside of their normal floating island of hyssop environments within the Titicaca Lake, you take them to the civilization, and they get tuberculosis a lot more easily. So there's some compensatory mechanisms that when you take them away from their environment, trigger other type of diseases that, are kind of weird. So trying to understand these differences, not just from the genetic point of view, but also from the clinical, from the sort of disease history, and the deep feature characterization and measurements, how we correlate those so that we can begin to understand these associations could help us have tremendous value for future research. So we had 7 populations. Matses and Uros, they're the most indigenous. And by that, I mean, they've had the least degree of admixture with with Europeans. And as we get closer to the right, they're more, we call them admixed. They have been sort of more inbred or mixed with Europeans and some others. So if we start looking into the specific DNA mutations that are characteristic for each of these populations, we would want to use something called variant effect predictor. Okay, so the variant effect predictor takes the DNA of a patient and then it sort of adds a number of algorithms that tell you something about the likelihood of whether an observed genetic variation, an observed genetic marker is likely to be pathogenic or non-pathogenic. That's the main thing we're interested in. We want to see whether any specific change that we observe in the genetics of a patient is actually contributing to disease or not. And in order to do that, we use software such as the variant effect predictor, which I'm not going to demonstrate it because I was going to, but based on your feedback, I'm not going to, because we're going to make it more getting hands dirty with the data. So here you have a prioritization of the almost 13 million genetic variants that we observed in these 150 genomes from those 7 populations that I showed you before. So it gets like a funnel, right? So you start, and don't worry about this terminology. What I want you to see is 2 things. One, that from the 13 million variants, we predicted that around 1,200, okay, these are all the variants that all of the genetic variations of the genetic differences that we observe in 150 million, sorry, in 150 individuals scattered throughout Peru, okay? Now, out of those 13 million, 12%, 1.6 million, had never been seen before, right? Which is astounding, okay? 12% had never been seen before. And around 1,200 were predicted as high impact. In other words, they would disrupt a gene and produce functional deleterious consequences. Any questions so far? How does that work? Yeah, I haven't explained it. So it basically looks at whether a genetic difference is inside an exon. If you remember, an exon was the bit that actually encodes for a protein, right? So that's one factor. So if you have a variation that changes the resulting DNA sequence, sorry, the resulting protein sequence, then potentially that's a risk factor. Now, there are going to be some changes that do nothing, but there are some others that can completely truncate, disrupt, and basically make the protein completely dysfunctional or non-functional. That's what we call high impact. You've had this expression of mouse knockouts, or when you have a sort of organism where you basically break the gene and then you try to see whether that has any effects or not. Many genes, you break them, nothing happens. But some of them, you break them and then you are either non-viable or you have this sort of bad disease. So computationally now you can predict the likelihood for a genetic difference to cause disease or not. And that's what we did for the 13 million. Obviously, it's a very, very, very small percentage So the vast majority of the variations and the difference do nothing. But we all carry 100 of these such deleterious mutations. And sometimes that has important effects in our susceptibility to disease. Obviously, the fact that we are alive and even though we are carriers, that means they may be compensated or they don't do so much. Any questions? Okay. So basically, I'm just here. The point of these high impact mutations is that the vast majority, we have no idea what their function is. So we are able to predict that they are pathogenic based on our existing knowledge, but also based on our existing knowledge, since many of these variants have never been seen before, or simply we just don't have enough knowledge body of data, the vast majority, we have no idea what they do. Okay? So part of my research was to then really further understand from those 1,200 high impacts, prioritize those that were most frequent in this population of Latin American, of Peruvian, right? So here I am, if I remember, the top most frequent 23 out of those 1,200. So why are they 23? Because I just set the arbitrary threshold of the fact that they needed to be more than 10%. They will have to be at least present in 10% of the 150, and less than 1% present in the rest of the world. So in other words, I am selecting for those which are incredibly rare in the rest of the world, but they are highly frequent in the Peruvian population. In other words, we are selecting for those that are uniquely present there, likely to cause disease. And that's going to give us a much better understanding of the specific genetic mutational features that could potentially help us inform current existing medical genetics procedures or preventative interventions, really. Okay? So when we then look at those genes, right, there are 23 genes that have been affected, that we've prioritized, we've selected. And now I'm going to use software that clusters the functions that these genes are likely to be involved in. Shall I repeat that? So I took the highly frequent Peruvian rare global affected genes by these functions. And since those genes, I can actually identify the biochemical, we have some information about those genes. Yeah. And now I'm going to see whether there's some kind of clustering, whether there's some kind of dominant functional theme around this list of 23 highly frequent rare genes. And guess what? Statistically speaking, I have a significant enrichment for those genes involving infectious disease and immune response. I use a tool called, well, I'm not going to say what the tool is, but what it does basically takes from a database the functional terms associated to each of the 23 genes. And then based on those terms that have been used, it clusters them according to their function, their likely, sort of high-level function. And the funny thing is that the enrichment are precisely related to the issues that we identified historically when the Spanish came, i.e., susceptibility to infectious disease and issues with immune response. So in other words, I cannot validate this. This is just a hypothesis, but I can hypothesize that perhaps this has to do with that bottleneck that I showed you earlier. Yeah, we still have the residual effect of what happened 500 years ago. So what this means potentially is that individuals may have a predisposition to infectious diseases for these uniquely frequent Peruvian disrupted genes. And that means that they could have, for instance, higher prevalence of certain infectious diseases, and highlights the need for targeted infectious disease surveillance, prevention, and treatment strategies. So genes involved in the immune response are significantly overrepresented in this subset of highly pathogenic, highly frequent Peruvian genes, rare from the rest of the world. Therefore, this could have implications in the way populations from Peru respond to infections, vaccines, and autoimmune diseases. I'm not going to go into this particular detail, but basically, because this is quite detailed. But here you can see that we then expanded from 7 populations to 28. And we then looked and performed something called a principal component analysis, which is an unsupervised clustering using genetic data. And from these 26, so for these 28 populations, we can actually see the geographical groups. So we can see how the populations that are in the Andes tend to cluster together. The populations that are in the Amazon tend to cluster together. The Afro-descendants, they tend to cluster together. The ones that are in the coast, they tend to cluster together. And then you have some of them which are admixed, very admixed, like the ones in Lima, in the capital. Okay? And again, you can see that just by looking at the genetics without even in a sort of blind way, you get this clustering, which makes sense when you look at the geography and the history. And then another bit of research that we performed is that we then combined not just the genetic data from these 28 populations, but we combined them with data from an additional 130 odd populations from around the world. So we use something called the Simmons Genome Diversity Project, which basically has around 200 many different populations, right? From around the world. And now I'm superimposing my data onto the rest of the world data. And so you can see that the Afro-descendant individuals from Peru, some of them tend to get really close to Africa. So here we have For instance, Luya, Laka, Yoruba. Are you Yoruba? So there you are. That's you. There. Oh, you cannot see really well, but you are here. Do we have Kasach somewhere? Maybe around here. Here it is. Uyghur. I'm sure you are somewhere here. Tibetan. Greek. Czech. So the way it works is that you have 20,000 genetic variants for, in this case, 1,000 individuals. These 1,000 individuals come from different parts of the world. And so you are reducing the spectrum of all the different genetic variants. So all the individuals, we test the same 20,000 points for all the different populations. So 20,000 points would be 20,000 dimensions, right? Because each point is one dimension. But here, what we are doing is statistically compress those 20,000 dimensions into two, and then maximizing the distances between any given individual by reducing while reducing the 20,000 dimensions into two. That's what the principal component analysis is. So you maximize difference while reducing the dimensions. And so this is kind of a projection of that reduction of 20 dimensions into two, while maximizing the distances between any given dot. So any given dot is an individual. Okay? So you guys see here the Peruvian populations, but then you see that the closest to them are Eskimos. And then you go a little bit further, you have Japanese, Alec, Shigo, Han, Bengali, Papuan, and then you get into European, Turkish, Absetian, Makai, Punjabi, and then you go up there, Sahrawi, Mossabac, and then you get Africa, right? Hadza. So, which I find fascinating because this allows us to really understand agnostically human differences just by looking at genetic differences, and that can help us, learn things about our patients, right? Any questions? So this is an example of what you can do with a biobank, but of course, this sort of biobank initiatives have been happening now for a while. And there's been a kind of population scale national biobanking project race where, the first national biobank happened in Iceland, of all places, in 2003, with a company called Decode. They come from an ex-Harvard professor who decided to go back to his country. And because Icelandic are very good at keeping records of the genealogy, so they actually have pretty much the tree from when the first settlers came and what they died of. So you can then correlate the appearance of diseases in the tree, extrapolating them from what you see in the current population. So you can see the different diseases in the current population, and then you can infer based on the trees when those, if any, actually arose in history. And they're going to have specific diseases because they've been a quite isolated population. Again, another bottleneck. The second one to really do something was the UK 10K. So the UK then did the first 10,000 whole genomes in 2008, which, considering the fact that the first human genome was released, just the first one in 2003, that by 2008, the UK was developing 10,000, that was a tremendous achievement. And since then, the UK has been leading the way. In 2013, they released David Cameron, the proposal that the UK was going to sequence 100,000 genomes, which was an absolute revolution. And you can see that they would invest 300 million pounds in 2014. And then the US said, oh, well, we need to catch up. And then you had these other countries that decided that, they were going to invest. So one of them was the Precision Medicine Initiative Cohort, which has changed its name today to All of Us. So some of you may have heard of that biobank, which is called All of Us. But it hasn't had the same amount of funding as Genomics England, as the 100,000 Genomes Project. And then you have some other biobanks that have proliferated. In 2016, Professor Dame Sally Davis released a very influential report called Generation Genome. In this Generation Genome report, the UK government committed an extra 250 million to continue with Genomics England. And then after that, we've had Genome UK, where the investment into genome for the whole population has been continued, and it will be continued. And as of today, the big initiative, which is now being managed, is called Generation, I think it's called Generation Genome, whereby as of now, they are sequencing 100,000 newborn babies across the whole of the United Kingdom. Together with the UK Biobank, which, as you know, now contains 500,000 whole genomes. We estimate that this must have cost just the whole genome sequencing around $50 million. Yeah, that's just the genome. And then the next big thing that is now happening is called Our Future Health, whereby they basically have the genomes, the clinical histories, and the many measurements questionnaires for 5 million people. That is 10% of England's population. And nothing like this exists anywhere else in the world. Any questions? Sorry? How is this going to work? Yeah, so if you are like, I have received a letter from the NHS asking me to enroll Our Future Health. So the NHS has sent millions of letters to people, and they have like vans. I got an email last week saying that Our Future Health van would be in my area if I wanted to go and donate. So there's a lot of recruitment effort behind this. Any more questions? Are you sure? There's something in between there.
You mean like donate what? Like you're going to donate your blood?
Yeah, so they take peripheral blood, And then obviously they take some, what do they call it? Anthropometric measurements. I think that's the correct expression. They, they take your weight, your height. They might do like a little questionnaire. And then with that blood, then they do a biochemical analysis. And then they take white blood cells. Yeah, they have DNA. And so from there, they run, they call it genotyping. So they run the sort of genetic tests. And then all of that is integrated with your NHS record. The UK Biobank, first of all, obviously size. And then the UK Biobank was more on the sort of voluntary basis. As a consequence, the people who were recruited are more likely to be white, rich, healthy, highly educated volunteers. Our future health is more targeting underrepresented populations. They go to where least well-represented communities are. They have a lot more engagement. And also, whilst the UK Biobank had a more sort of epidemiological focus, that is understanding disease really as it is in the UK. Our future health is more on the predictive side of things, and they're implementing, for instance, models that help predict your genetic risk for diseases, and if they find something, they will contact you. So the UK Biobank is more on the current epidemiological understanding. Our future health is more on the preventative medicine. Any more questions? Okay. So now I said that for the assignment, the first assignment for biobanking, for data science, you will have to have groups where you guys basically provide a fake proposal for a biobank, and as a group need to think of the different components of the biobank that needs to be taken into account, i.e., recruitment, ethics, things to do with the design of the technology. How would you go about it? I'm not expecting you to really cover every single base, but at least think about the requirements, and then being able to present this to the rest of the class so that you can think about the principles behind these resources, which, as I said, pretty much all of the biomedical discoveries, to some extent or another, use this type of data, right? Even if not directly, they do it indirectly. The UK Biobank is kind of usually the sort of validation or the reference data that people use for validating their research. So I'm going to introduce to you the 50,000 genomes per root. Yeah. And this is a mock presentation of this data bank, right? So that gives you an idea of the type of thing I would expect. You can take it as an example of the type of presentation, right, that you could give. So you need to present the overview and objectives. So the objective is to develop a national study in Peru mirroring another data set biobank called Regenerons, which basically was performed. This is a real one. It was performed in Mexico City. They used 50,000 genomes, and it was used to characterize the genetic diversity and disease susceptibility, as well as protective environment for populations. In this case, we want to look at the environmental protective genetic variants within the Peruvian population. So what's the scope? We're going to include sampling from across 14 regions of Peru for broad genetic representation. And we want to chart the genetic diversity of the coast, the mountain, and the Amazon environments so that we can address the endemic conditions for precision medicine approaches in Peru, which, as I said, precision medicine approaches, which are based on big data, do require that the population you are going to give precision medicine approaches have been represented in existing models. Otherwise, it's not going to work very well. So we want to build for this a reference set of genetic differences that could help us identify those genetic bits that are clinically relevant for these populations and nowhere else, with the subsequent objective of building capacity, because there's not a lot of infrastructure or skill development within the region. So there's also this sort of the more benefit sharing aspect is to help Peru to acquire infrastructure that would lead them to have a resource that they can use, they can learn from, they can develop. So what are the components within the study design? So we will have to think about the ethical approval, about the protection and compliance of the data, material. In terms of legal, we will need to think about the material transfer agreement, have a memorandum of understanding. Then in terms of sample collections, you will want to think about community engagement, participant recruitment, sample extraction, then how you're going to handle the samples, how you're going to store them, how you're going to develop an infrastructure to hold the data and make sure that that data serve the purposes of advancing the health priorities for the community and in a global stage as well. You also need to think about the nurses who will be taking the samples, the researchers in the local area, the actual principal investigators, the travel needs, and then all of the technical infrastructure behind the development of the sequencing and the production of all of the biochemical tests around it. But then also how you approach public relations and dissemination of results. Okay, so this would be all of the different things that you would want to think for a biobank. Any questions? Do you agree? Yeah, is this useful? Okay. Yeah, it's already up. Any more questions? Any more observations? Any more questions? Tunde. Pardon? Sorry.
Sorry,
Recordings to know those rights.
what's on Teams? Sorry,
Recordings for this.
recordings. Oh,
Yeah. We're being recorded and transcribed. Okay. So we're going to involve 21 universities, 16 of the 24 regions within Peru. You have them here in color. And then native communities. Now, what do we mean by native communities? And this has to do with the sample selection criteria, okay? So we mean by native communities, individuals with parents and grandparents born in the same community, speakers of a native language as mother tongue, with distinctive native cultural legacy and not a mixture with other communities for millennia, and the possibility that they can be extinct due to, for instance, due to climate change and migration, because economic opportunity. And that's something that we found, that many of the indigenous populations, as soon as they get in contact with the civilization, they emigrate because they want to have better economic opportunities. So then they'll go to the capital. I guess that will happen in Nigeria as well. Like most people from the countryside, they will go to Lagos or Abuja, or because there are more jobs, and they want to have better healthcare, and they want to have, some prosperity. So how would we share with the locals? We would request that 4% of the royalty share of all benefits that are derived from this data that are shared with the local community. That they also provide that, whoever, let's say pharmaceutical companies, that they provide compute facility maintenance for 10 years and support for governance and ethics, community engagement, sample collection, blood testing and analysis, capacity building, and the salaries of the groups. So what do we give in return? So we give access to unique and underrepresented data, opportunities for new drug discovery and target validation, especially for diseases prevalent in Latin American populations, including type 2 diabetes, cardiovascular diseases, and tuberculosis and dengue. Also, participation in data analysis and scientific research, as well as intellectual property, enhance global brand and corporate social responsibility, as well as improve models for prediction of risk in genetic contributions to disease, as well as strategic partnerships and market expansion to those specific regions. And we've calculated that all of this will cost only $50 million. So give us $50 million, and then we'll give you all of that. And you'll feel good about it. So what are the estimated costs? So we have these categories. So in terms of governance, we have the breakdown ethical review board operations. Then we also have sample collection and startup that includes sample collection, participant recruitment, consumables, blood sample analysis that includes blood cell count and hematological biochemistry. Then you have sequencing, data storage, and then analysis and research. So you have the bioinformatics, software, compute resource, data transfer, cloud infrastructure, and research support, community engagement. I'm going a little bit fast, but now you can start to see all of the different components, which I think, kind of opens your mind to understand what's behind these data resources. Yeah. Big pharma can give, they have big pharma, for instance, or governments. At least that's what I'm hoping. Any more questions? You have some details there, and you have a Gantt chart of, the different main stages, how long it would take. So we did this for 5 years. And so what would be the next actions based on this? So, the next action, once we have this plan, would be to confirm goals with potential Companies establish a steering committee, logistics, data collection, processing, analysis, and report. Now, let's talk about some specific issues here. So, have you heard of data sovereignty before? Let's discuss that. Yeah, we're in the 21st century, right? So, data sovereignty. What do you understand by data sovereignty? Do you think that that particular buzzword would be good to know what it is? And why do you think it's important? Yeah, who controls the data? Who have access to the data?
The government.
It could be the government. I mean, that has to be agreed, discussed, put in a contract. So I'm just sort of explaining the things that we need to consider, right? We need to consider data sovereignty, which usually has to do with the people that that data belong to, actually. So how do we protect? How do we make it that they benefit? And how do we make sure that data benefits communities, that communities approve the uses? Yeah, once you give that data, there's no way for you to control what is being used. So you need to have the appropriate safeguards. Accountability for data protection and transparency, as well as making sure that the usages of that data are aligned with the local people's values, yeah, and international law. So there are international data protection compliance. For instance, in the EU, we have GDPR. I think I've mentioned this before. So GDPR is paramount, basically, and it's not just for Europe, it's global. You know, even Google has to, and all of the big, magnificent 7 companies in the US have to abide, because it's just such a big market that, and they sort of set the standards. And GDPR stands for General Data Protection Regulation. It basically says that if you are going to use any identifiable data from anyone, you need to have that informed consent. That's what it comes down to, basically. And that's why, since you have GDPR in action, that's when you started getting having to choose whether you want to accept all cookies or decline, or yeah, that was the trigger. Any questions? In Peru, you had the National Personal Data Protection Standard, and then in the US, you have the Genetic Data Sharing Protection. So you have the San people present of South Africa that receive 4% royalty of on commercialization. And so in our case, we would want to follow the same example. And that basically allows to have true partnership. This is what I like, reciprocity. Okay. Any questions? Any comments? Do you agree with that? Do you disagree with that? Is this important? Why is it important, Catherine? Yeah? Yeah. We make sure that we act in an ethical, responsible, transparent manner, right? As any researcher nowadays. Yeah. Now, so there have been some research findings, which I'm not going to go into details today. But there's also a very interesting thing that I think I can offer, the biobank design for resource constrained settings like this one, right? Most of you come from the global south, maybe I could say, or maybe not, but in some countries, there are key challenges, right? So in Peru, for instance, how do we make sure that the storage and logistics for sample transportation from one place into the hub happens in a way that samples are not mangled? You have to store them at minus 80 degrees centigrade. That's not trivial. Yeah, if you have to bring them from remote places. Also, the fact that there are fewer trained personnel in data handling and higher costs. That's kind of counterintuitive, so it's actually more expensive to run stuff in cheaper countries simply because they have to bring the technology. So what costs here X in Peru will cost X plus N because they have to bring it from abroad, and it can even cost a lot of the time double or triple. And a lot of the time there's not even supply to those places. Yeah, some of those genetic tests won't be sold there simply because the company just doesn't have business case for those. Would you agree with that? Is in what happens in Guatemala? That some of the things that are sold here are not sold in Guatemala. And that's something that we forget here in the global north, that we have access to be able to buy anything, but there are a lot of restrictions. Even just bank account paying, they take these massive commissions that you wouldn't have here. There are many more barriers. I mean, maybe in Peru that might not be,
In Guatemala, it's like that. I've had really good bands.
So many people might not even have a bank accounts. They might have a mobile cell phone, but they won't have a bank account, for instance, right? Okay, so let's have a little bit of discussion, shall we? That's all I wanted to say. And now I would like to stop sharing. I'm going to stop recording as well. And hopefully that gave you a little bit of a taste of the amazing things that you need to think about. Yeah.